All right, good morning. Thank you all for coming out for the first session of the day. It is early for us, just as it is for you, so bear with us. I am Jonathan Mosley. I'm very happy to welcome you to our workshop on titled Moving Towards Consensus. So we're going to be talking about best QA/QC practices in LC-MS-based metabolomics. And just to give you a little bit of background, we are all from an organization called the Metabolomics Quality Assurance and Quality Control Consortium, or mQACC. And mQACC was formed a few years ago now, following a think tank that was sponsored by the NIH's National Cancer Institute in October of 2017. And the consortium was formed specifically to engage with the metabolomics community so that we can communicate and promote the development, dissemination, harmonization, et cetera, of best quality assurance, quality control practices, because this was a huge area of where there was a gap in information from what people were doing and reporting in the literature. So currently we have over 100 members from 4 continents, and they're representing a variety of academic, industrial, and governmental organizations. And if you are interested, there was a white paper that was published in Metabolomics in 2019 that described both the think tank and the formation of the consortium. So before we really dive into it, we just want to lay it out for you what we consider quality assurance and quality control. So we define quality control as the processes related to the procedures applied during and after data acquisition. And then quality assurance are the processes related to the procedures applied in preparation for data acquisition. However, rather than putting both of them sort of in the same timeline, you can see in this bubble plot here, you can have activities that happen before, during, and after the data acquisition process, and then below that, you have quality assurance. So it's kind of happening all the time. And the objectives of what we're going to get into today, to put it in the context of the bigger picture. So we are here to disseminate our findings of the best practices working group. We have been engaging with the community for the last few years, and we have been gathering information related to best practices for LC-MS-based metabolomics. And we've been doing this according to 8 key areas. So if any of you were in Valencia last year, we held a workshop that covered 4 key areas. Those are the 4 that you can see highlighted in the top box. Today, we're going to focus on the rest of the story. All right, so those 4 boxes on the bottom are going to be the topics that we covered today. All of this information is being gathered, compiled, and assessed for the emergence of a best practices living guidance document. So this will form the content of this guidance document, which will come out and be publicly available. It's living, and we'll talk more about this too and later, so I don't want to spend too much time on it right now. But speaking of what the next 110 minutes are going to look like, we have our 4 topic speakers and presenters up here. So we're going to have Rick Dunn all the way on the end, Matt Lewis, second, third is Tracey, and fourth, Dajana. Now, they're not going to get up in that order, but they are all going to get up one at a time, and they're going to present to you each on one of these topics, and then we're going to have a time of discussion that will be guided by a single polling question per topic. I lied, we have 2 for metabolite identification, but that's okay. We're going to get through this. You guys are going to have a great time. We're going to interact with each other, and hopefully after this, you guys can just go home because you will have done what you needed to do. Just kidding. All right, real quick, just to acknowledge everybody that has put effort into this. This has been a lot of people working for a long time. I'm not going to read all the names, but I will just point out that we've got a great group of people in NQAC who are all very, very good workers, very easy to work with, and they all deserve to be acknowledged. With that, I'm going to ask Rick Dunn, our first speaker, to come up, and he's going to talk to you about our first topic.
Great morning, everybody. So I'm Rick Dunn from University of Liverpool, and I'm going to talk, for those of you that know me, metabolite identification and annotation is a key topic in my research, so it's great to be up here and talking about this. So we're going to talk about metabolite annotation and identification in LCMS-based untargeted metabolomics. We're not going to talk about NMR or GCMS. That's something that will come in the future within mQACC. And most of what we're going to talk about today was based upon polling in a workshop that was run, online workshop that was run about 18 months ago. So just before we go into the polling questions and give you some information about those, some of the results, let's just think about, there's two terms that are commonly applied in metabolomics, annotation and identification, and they're used interchangeably, and sometimes we're not quite sure about what the definitions of each of those are. So I wanted to talk about both of those. And really they are about confidence that you have in those names of those metabolites that you provide within there. Identification, you're typically comparing two or more complementary types of data to data that's been collected for an authentic chemical standard, and that data's been collected in your laboratory applying the same assays that you apply. That's identification. So, for example, you're collecting and you're using, for example, chromatographic retention time data, MSMS data, and you've run an authentic chemical standard and you compared the retention time and the MSMS spectrum for the chemical standard and in your biological sample, and you're getting a match under certain criteria. So that's high quality. Okay, high confidence within there. Most of actually what we're doing is annotation, okay, and there's different levels of confidence within annotation as well, but annotation is typically where you're using maybe two, maybe one different type of data within the LCMS. It might be retention time, it might be MS1 data, MSMS data, and you're matching it to databases or to mass spectral libraries or retention time libraries, but they haven't been created in your laboratory, okay? So, for example, you could be using MSMS data and going and searching MZ Cloud or Metlin, etc. within there. So that's typically what we define as annotation. Actually, most of what we do in metabolomics is annotation for various reasons based on these definitions. Now, I'm just going to use the term identification during the course of this short presentation, but we're including annotation identification within that. So I'm realising that I need glasses to be able to read the screen down here, so I need to go to opticians later. But the first question is, what type of data do you use to identify metabolites and choose all that applies? So these were polling questions that were applied within there. We've got different examples within there. We've got MS1 spectra, we've got MSMS spectra, and various other ones within there. We've got retention time as well. And importantly, and as you'd expect within there, it's the MS1 data, so the exact mass of the intact metabolite. It's the MSMS data and its retention time data that's been most widely used for identification of metabolites within there. And I think everybody that's using LCMS would agree with that. Ion mobility is being used as well within there, and MSN for some of those, for instruments that can apply MSN as well within there. So that's good. That's what we'd probably expect to be within there. So the second question was, what mass accuracy criteria at MS1, so not talking about MSMS, but just at MS1 level, do you use during your metabolite identification? And as you can see here, so the numbers along the top, I should say, are the number of responses, not a percentage within here. And as you can see, the majority are using up to 5 ppm, okay? There's sort of more than the majority within there. Some groups are using, some researchers are using sort of 5 to 10 ppm within there as well. There's not many groups using over 10 ppm. So the majority of the community are using 10 ppm or less, and actually a lot of those are using 5 ppm or less within there. So that's interesting to know for the criteria that we should be thinking about using when we're identifying metabolites. So the third question was, what type of retention time and MSMS libraries do you use to annotate, to identify metabolites? And again, choose all that apply within here. And then the first three questions here are based on MSMS libraries, the first three options, and then the next three options are based on retention time libraries. And you can see for MSMS libraries, remember again, it's number of responses, not percentage, along the top. You can see they're using both in-house libraries, but also publicly available libraries as well. So they're using a mixture of those, so using both of them basically within the identification within there. Some are using MSMS libraries, so in silico-based libraries within there. So for example, GMPS is one that comes to mind for me. But for metabolite libraries, actually the majority, by far the majority, are using in-house libraries. And there's a very good reason for that, in that MSMS libraries, you can collect data you can collect MSMS data in a laboratory and it's transferable to other laboratories because we're using very standardized parameters for that. For example, the collision energy can be defined within there. But for retention time libraries, because LC columns, LC methods, solvents, gradient elutions, etc., all of those change, retention time libraries aren't necessarily transferable between different laboratories. And so really there is the view that, at least at the moment, that everybody's building up their own in-house retention time libraries for doing that. So there's a difference there between MSMS libraries and retention time libraries and how we construct and how we use those. The fourth question of the poll, there was multiple questions. We've picked just certain ones out of those multiple questions within there. So how many metabolites do you typically identify with in-house MSMS and retention time libraries? So in-house, but also MS and retention time libraries within there. The most respondents, okay, for any one answer was 1 to 50, but most of those you can see it's going up to about 200, which I think is about right if you think about these and the amount of chemical standards that are available within there. There are some that can measure over 1,000. I was a bit shocked by that, I will say personally, because of the amount of standards that are available, but they're saying that, so that is possible within there as well. But we're seeing low numbers, low hundreds, basically, of metabolites that we can identify using those in-house libraries. And remember, we've got, for MSMS, you've got those publicly available libraries as well, so that will probably increase these numbers up as well. And then the fifth question, how do you confirm separate detection of isomers in your identification in the case of multiple isomers being present? Okay, and this is actually quite a common situation in untargeted metabolomics. There's a lot of isomers in there in biology. Thinking, you know, an isomer is everything that has the same molecular formula, so therefore the same mass that we're measuring in mass spectrometry, at least in MS1, but they have a different chemical structure, different structure of those atoms in space within there. So if you can think about all the isomers that are present, all the hexose sugars, leucine, isoleucine, and the list goes on and on and on. So these are some interesting ones within here. So I liked the fifth question down, or the fifth answer down, where most people will report all of the potential isomers. So with MS1, you can't separate between the different isomers. You've got to use chromatography and potentially MSMS, or a mixture or combination of those ideally to be able to separate out isomers within here. So most people were reporting most of those, which I think is good practice for doing that. The others were applying or looking at the MSMS data for the isomers and looking to differentiate between different isomers based on that. Sometimes that works, sometimes it doesn't. Okay, isoleucine and leucine are very limited MSMS mass spectra, separating those based on the MSMS mass spectra. Ion mobility, it was great to see within there, and actually quite a traditional approach is to use two different assays, so use two different chromatography assays and look to separate out isomers using that. So they might correlate, for example, in one assay, but in the other assay you'll see separation of those within there. And finally, so what quality indicators do you use to assess your confidence in metabolite identification? And so this is important. We'll come back onto this actually in the next two slides as well. So a lot of people were using a lot of different indicators, which is great. We want to show all of the evidence that we can and to identify the confidence that we have in that identification within there. So most people were using MZ error. I assume that's for MS1, but it could also be for MSMS or MSN data within there as well. They're using retention time, they're using the MSMS match score within there, and they're using the isotopic distribution. So by that we mean, for example, the M and N+1 peak and the intensity ratio of those to define how many carbon atoms are present in the molecular formula, for example. And some people are also using the fine isotopic structure, so that might be looking, for example, for specific atoms within there, between, say, for example, 32 and 34 sulfur, 35, 37 chlorine, etc., within there as well. So it's great to see a wide range of different complementary indicators being used, and I think that's really good practice for what we're doing in metabolomics. So this is important. I'm going to test you on this in a minute with a polling question. Okay. So there's evidence and criteria. Okay, so evidence is really reporting what you've measured and reporting that for your chemical standards, or if you're using an MSMS mass spectrum from a publicly available library for your chemical standard, but also for the metabolite that you've detected in your biological sample. So evidence, for example, could be the retention time of your metabolite in your biological sample, but also in a chemical standard in your in-house library, for example, within there. So that's given the evidence, okay? But there's also criteria. We use criteria to say whether we feel or we're confident that it's a match to that metabolite or not. So that's what we define by criteria. So that, for example, could be the MZ error or the mass accuracy. So are we using the 5 ppm, 10 ppm, etc.? Is it the retention time error? Are we, for example, using a retention time error of plus or minus 5 seconds? All of these provide the confidence within there. We can all report the evidence, but not necessarily understand how that's being applied within there. So criteria might be important within there as well. So polling questions. Okay. So let's introduce how to do a polling question. I get the lucky choice of doing this this year. So step one, if you've got the app, hopefully you've all got the app. Okay, who hasn't got the app? A few of you. Jennifer, oh, I'm shocked. Sorry, I'll pick Jennifer out because I know her well. So hopefully you've all got the app. So there's going to be a number of polling questions during this session, so keep your app open within there. So open up the app, select Agenda, and then select W9, so Workshop 9, which is this workshop, in the agenda. And then click on the arrow next to poll. And I know this works because I went in yesterday and checked that it does work. Okay, and I know Jonathan's checked it about 10 times in the last 2 days to make sure it's working as well. And then the page will remain blank until the polling is activated by the moderator. Has the polling been activated by the moderator? Yes, Krista's our moderator. Brilliant, thank you very much. So, vote away. Actually, no, you need to know what the question is before you vote, don't you? Sorry. So the question is, we've got 2 polling questions here. So the first question is, when reporting metabolite identifications, which of the following should be provided? The quality criteria only, the experimental evidence only. So remember, the quality criteria is what is the acceptable error, for example. So acceptable MZ error, mass error within there, retention time error within there. Experimental evidence, so what is the retention time in the biological sample in the retention time library? Should it be both the quality criteria and the experimental evidence? Should it be neither the quality criteria nor experimental evidence? Or I don't do metabolomics, so I don't report metabolite identifications. So vote away, and Chris is going to tell me when to stop. It's great to see so many people first thing on a Monday morning. Everybody found that easy to do? Yes, good. These are important. These do feed back into what we're going to do next, for example, and thinking about what we're doing next. Okay, so 20 seconds if you haven't done it already. This is really the first question, is just testing who's awake and who's not. Yeah. So these are important. We typically publish a workshop report. Last year's workshop report is under review at the moment, so all of these will be included in that as well. So it is really important to get your views on what we should be doing within mQACC. Yep, okay. And the second question, okay, is, so guidance for the quality of metabolite annotations and identification and targeted LC-MS-based metabolomic samples in upcoming mQACC guidelines should cover which of the following, and you can choose all that apply. So if you think all of them are important, apply for all of them. If you only think 1 or 2 of them are important, then only include those. So option A is considerations in choosing the quality criteria. Remember the MZ error, retention time error, MSMS match score, minimum match score within there. The types of evidence, including use case scenarios. So, for example, the retention time information for biological sample and your chemical standard. The evaluation metrics, reporting guidelines. Others, okay, you don't get to include what those others are. And then I do not think that guidance for this topic should be included. And Jennifer, you've got your hand up. Is that the same for everybody else? Okay, so choose which you think is the most important. So apologies for that. And when we're done that, I'm just going to get you to do a hands up poll very quickly. So let's do a hands up poll anyway. Okay, so hands up if you think considerations in choosing quality criteria is important. Yep, good selection. Yep. Types of evidence, including use case scenarios. Fewer people. Okay. Evaluation metrics. Quite a lot of people. Reporting guidelines. Thank you, because the Metabolomics Society are now working on these, so it's great to see that you want those. Anything else? And you can ask this in the questions. So if you've got another, then let us know. So of those, whoever's making notes, the considerations in choosing quality criteria and the evaluation metrics are probably the most answered. And then B and D were probably about 50–60% of those. Yep, okay. Okay, we'll close that down. So now we're going to move on to Tracey. Oh no, we're not going to move on to Tracey. Sorry.
All right. So before we jump into the next part, we do really want you guys to be able to choose all of the options that you think are important. So what we'll likely do is at the end of the entire session, we'll re-ask that question where you should be able to answer everything. But we can still discuss what we have done so far. And this is the part where we ask the polling questions, we get the data, but you can only get so much information in a polling question, and we want to have as much relevant content in this living guidance document as possible. So this is your opportunity to share more detailed information about what you do. I will ask a question. We have two lovely assistants who are going to be walking around with microphones, Lise and Carol. So if you feel like you have a response to my question that you would like to share with us, then you just raise your hand. They will bring you a microphone. We have two, so we can kind of go too deep. And we're going to give ourselves about 10 minutes to discuss, go back one more, this question and the responses. And I'm going to ask a follow-up question. And because it's the first thing in the morning, I've got a special treat. Whoever answers the first question is going to get a perfectly packaged and sealed, just a little squished, blueberry muffin, okay? All right, so the question that I'm going to ask is, did you share the results? You also, by the way, should be able to see in your phone the results of what you just answered to this question too, okay? All right, so what we want to know regarding in-house libraries, how are your libraries built, okay? So we want to know which standards do you use, how are they measured, collision energies that are chosen, et cetera. So for in-house, specifically MS, or MS to the N libraries, how are they built? And if you have a microphone already, you can stand up and talk. If you don't and you like one, please raise your hand. Remember, there's a blueberry muffin on the line. All right, we got one over here. Well, then it's just an added bonus.
Good morning, everybody. My name's Arpana Vania, and in the Fiend Lab, I'm considered as the librarian, so, it's a good question. When we're building libraries, we usually use mixes of standards that are purchased from different vendors. And we usually have about 20 standards, 20, maybe 25. And we run them normally at a 1 ppm concentration, because that's when we see like good enough signals to get really good MSMS data quality. And depending on the instrument that we're acquiring data on, let's say, for example, the Thermo mQACC, we'll do stepped collision or normalized collision energies of like 20, 30, and 40. In previous experiments, I usually do PRM methods or different ways of acquiring MSMS data at different collision energies, because then when you're processing the data, you can actually separate the different collision energies. So I do have a lot of interest. Like I said, I'm the librarian of the lab. So if you wanted to discuss how to acquire library data, I'd be happy to talk about that.
Awesome, thank you. All right, I saw a hand on a microphone. Jennifer.
Yes, so we have two ways with LCMS. The first is very similar, using stepped collision energies. The second is that we often move standards that we've developed for targeted pathways into our untargeted. And that's really nice because we've often spent quite a lot of time in optimizing both the collision energies and which our reporter ions should be. It's early in the morning.
All right, thank you, Jennifer, and thank you, Arpana. All right, anyone else would like to answer this question before we move on? Yes. Microphone is on its way.
Hello, this is Marin Kolashevska. So I'm Thermo Fisher Scientific representative. I'm responsible for MZ Cloud, so I can share how do we do the in-house library, let's call this like that. And we used to take the analytical standards, cover the both ionization modes, and go into the MSN until it allows us to go. So it's in 3, 4, 5, 7, 9, depends where we can actually stop, what's the limit of the visibility in the machine. And we cover usually a wide range of energies. So starting from, let's say, 5 to 100, every 5 or 10 depends. That's how we do. Thank you.
Excellent, thank you. Okay. All right, unless we got anyone who really wants to answer that question again, which we're more than open to, I can ask another. All right, so for the first option, if you're looking on your screen, considerations in choosing quality criteria, okay, seemed to be a popular response. Here's our follow-up to that. What are the appropriate criteria for determining the quality of metabolite identification? So we've identified that it's important to have considerations in choosing quality criteria. We want to know, what do you feel are appropriate criteria? Yes. Microphone's coming.
Hi, my name is Catherine. I'm with Liverpool Sigma. I feel like it's a little bit obvious for the first one that it's going to be mass accuracy, right? So ppm thresholds, I think, are very important. In addition, I like to have at least 2 confirming fragmentations, so 2 fragments that match the database, not just 1 in the parent, but hopefully 2 actual fragments. And then I also like to have technical replicates of that. Some people don't think that's necessary, and that's fine. But I think it's your system should also be evaluated to make sure you're actually getting those fragments reproducibly. So that's mine.
Very good. And being the first one to answer that question, you have the benefit of the low-hanging fruit. So That's perfect. And it's okay if you feel like what you do is not what other people do, that's okay, because we're trying to gather the breadth of what is being practiced in the community. So we want to know everything. All right. Yes, down in front.
Yeah, this is Phil Brits-McKibben from McMaster. Just to add on to that suggestion, I guess the higher level of confidence would even to have a rank score function if you compare an analytical standard to your unknown compound by characterizing the MSMS, not only in terms of 2 or more diagnostic fragment ions, but also the relative intensities. Yeah. And coelution. Yeah.
What was the last part?
Coelution
And
with
coalition,
the retention
of
time.
course. Yes. Awesome. All right, we have time for one more response. Over here in the orange.
So this might be a different way to think about it. So before we actually go ahead and do any type of annotation identification, we actually look at the quality of the actual feature changes, right? We first identify, what's the apparent fold change, what's the CV, how consistent that feature is across the multiple different groups. So we don't bother annotating until we have something that seems to be a statistically significant change.
All right, excellent, thank you. All right, we are unfortunately out of time for this discussion, but don't worry, we're gonna get to discuss a lot more as these other topics come up. So right now, I'm going to bring Tracey up to the stand. She's going to talk to you about our next topic. Thank you guys, that was a very lively discussion, and let's keep it up for the next topic when we get there.
Okay, good morning, everyone. So this is an exciting topic. We'll talk about our use of reference materials in LCMS-based metabolomics, untargeted metabolomics. So the forum was, our forum was in March of 2022. And to set the topic, we are not talking about general quality control materials. We're talking about a reference material. And this is a homogeneous, stable material that's well characterized for a single or multiple chemical and physical properties. So an example of a reference material could be a certified or a standard reference material, which has the highest degree of traceability to an exact mass of an analyte, a pure analyte. Another example could be other commercial reference materials or a long-term reference material, and that's more of a purpose for a reference material. So just to restate, we're not talking about pooled QCs or blanks. So we first wanted to assess the community for the composition and the source of their reference materials. So we have biological reference materials, which include a matrix, and a matrix is important for an MS analysis. You often have matrix effects. And then we have synthetic reference materials. And it seems that the community is largely sourcing these commercially. Some folks are building in-house reference materials biologically, and then on the synthetic side, a lot of folks are doing a mixture of commercial and in-house, adding their specific analyte that fits the purpose. So we then ask the community for examples of the different reference materials that they're using. So the biologically based materials often are the NIST SRM 1950. This is the only SRM that is available for the metabolite, certified for the metabolomics community. And then other entities are, you can purchase materials through Sigma and BioIBT. And if you don't know BioIBT, this is a source that collects materials for research purposes. So it's a good source to build a reference material. On the synthetic side, folks offered Avanti polar lipids that they're often using. And then some folks are using a mixture of standards that they make in-house. And then you can combine both biological and synthetic materials. For example, the IROA technology, which are uniquely labeled internal standards, and you can add them to your favorite matrix, like a yeast extract. And then other folks are using SRM 1950 and amending them with a synthetic standard that fits their purpose. So I would like to invite you to check out this review article. It was recently awarded the 2023 Best Review Award for the Metabolomics Journal. This was put out by the Reference and Test Material Working Group of mQACC. And basically, it's a catalog of the reference materials that are available for the community. So then we wanted to ask about important characteristics and purposes of your reference materials. We asked folks to rank the characteristics that are important for them when they select the reference material. So the winning score was for a stable material. Stability and homogeneity go hand in hand. We're looking for a material that's the same over long periods of time, years, SRM 1950s at decades now. Also, cost and availability is important because we want a large community to be able to access these tools so that we can enable data harmonization. And then purity, traceability, and uncertainty are very important in quantitative analyses. So then we asked for what purposes are you using your reference material? And it's quite evident that there are multiple purposes that folks are using in their laboratory. Quantitation, normalization, and identification. Folks are using reference materials for instrument precision and instrument readiness, so a system suitability test. Less frequently, people are using a reference material to compare across laboratories. But this is an important concept, especially in multi-center, large cohort studies, to be able to bring the data together. And then in our discussion, other purposes came up, like a training material for newcomers to metabolomics, and also retention time prediction, which will aid in metabolite annotation. So how are reference material used and evaluated? We asked, do you use reference materials to assess your data quality? So for example, sample variability. We asked, do you use reference materials to correct your data quality? For example, batch corrections. We asked if you use a single reference material to both correct and assess, or do you use multiple reference materials to both correct and assess? And it looks like the majority of folks are using multiple reference materials for multiple purposes. So I wanted to highlight an example of a reference material use here. This was a group of lipidomics scientists who wanted to look at the effect of the mass spec introduction method on lipid quantitation. They used 2 commercial pooled plasma samples. One was SRM 1950, the other was another commercial material. 3 introduction methods, HILIC, reversed phase, and direct inject, all on the same mass spec using the same internal standard mix, and for the analysis of 75 lipids. So their initial data analysis showed, yes, there are 2 different plasma materials, but there's also an effect of method. So they decided to normalize their data to SRM 1950, which has community-sourced reference values for these 75 lipids. And they were able to correct for the method differences so that we're only looking at the biological difference of the samples. So data comparability over time and across labs is quite important for data harmonization. So then we asked folks, what criteria do you use to compare your data from reference materials over time? And the top 3 were mass accuracy, retention time shift, and peak area, peak height. The other criteria are also important to consider if you really need to dig deep in your data to be able to assess whether the data quality of your run is valid. Then we asked, where do you inject your reference materials? And the large majority of folks are injecting the reference materials both at the beginning and the end. And this follows the model of increasing cost, lower frequency. So SRM 1950 has a higher price tag, and people are using it less often in their everyday analyses, but it's important still to include a couple. And then other folks are responded that they are using reference materials in conjunction with their QC materials. And we think this is a very a great strategy to consider, and I'll show you, highlight a case study right here. So this is a group from, I think it's mostly Georgia. I think Georgia Tech folks are in here too, doing C. elegans work, and they really stack their batches with quality control materials. So they start out with a library standard, then a phenotypic pooled QC, a control pooled QC, a matrix match reference material, a blank, and then the reverse is done at the end of the batch. And in the middle are their QCs and their test materials in iterative batches. So this was a 6-batch run, and they used strict criteria where only 100% of the features detected in QCs are analyzed. But in batch 6, they had very few features pass. All QCs failed across all samples. So they were able to leverage the pooled quality control samples and the reference material matrix match because they were of the similar composition and they were in multiple locations within their batch. So they were able to conclude that their test materials were okay, their quality controls were okay. So redundancy in QC strategy is important. From our information gathering sessions, we've learned that there is low reference material use in the community. Oops, that's a little wacky. So we asked, what are the obstacles that are inhibiting your reference material use? Some folks requested more certified materials and different matrices, like mouse organs and more plant species. But just to remind you that with increasing certification comes an increase in cost. I'll go back. Oop. There it is. That was weird. And if you want to have certificate in metabolomics, we don't want a single analyte that's certified. We want multiple analytes that are certified. So that increases also the production time of a material to your door. For standard mixes, there's not a one-size-fits-all for all studies. And so most people are mixing and matching different mixtures. This causes a little bit of difficulty in solubility across the chromatographic range, so something to consider. So we think it's a good idea to have more discussions on reference material topics to enable more folks to want to use reference materials for high data quality. So just to wrap it up, reference materials serve multiple purposes within metabolomics workflow. The composition and characteristics define the application. Injection frequency and placement are specific to your purpose. And if you're using reference materials, we encourage you to provide the data, how you evaluate it, and what are you doing to your experimental sample data. So that brings us to our polling question. Is that up? We ready? Okay. So the use of reference materials in untargeted LCMS-based metabolomic studies in upcoming guidelines should cover considerations in choosing a reference material, B, purpose descriptions, including use case scenarios, C, injection frequency and placement within a run, D, evaluation metrics, E, reporting guidelines, F, others, or G, I do not think guidance for this topic should be included. So we're using reference materials, and you want to know about considerations in choosing a reference material. Why do we use a reference material, and how do we use a reference material? When should we inject it? how should we evaluate it? And let's report it, please. Okay, one more minute. Sure. Okay, 30 more seconds. Last chance. Okay, now Jonathan is coming up to help us discuss this topic.
All right, thank you so much, Tracey. Okay, you should be able to see in your app now the results. So, looks like a pretty even split amongst those first 5 choices, which is very interesting to me. That means all of them seem to be important. So, okay, great. I'm going to ask you guys a follow-up question, and we should have our microphone runners are setting up in the back here. So, here's your question. Do you think that QC redundancy is important in your study? Why or why not? So, yes, that is a yes or no question, but we want more than yes or no answers. So please tell us if you think it's important, and if so, why? If you don't think it is, give us some examples of why it's not. For your study, okay? Remember, this is specific to what you are doing. All right, we got our first question down in front.
Yeah, I guess in our case, we have a large cohort of, say, fasting serum samples. So we do use the SRM 1950 plasma pooled samples. So it's not quite the exact same matrix or blood specimen type. So we also include a pooled QC from that particular cohort, but do both. So that's sort of the redundancy option we consider.
Do you have any specific examples of when that's been important during a study?
Yeah, I guess for certain metabolites, there are some differences in, I guess, reported concentrations and abundances, so between serum and plasma, so there's some discrepancies between the specimen types, but plus, as you mentioned, those samples from this were collected at a certain time period. They might not reflect, say, exposures that are more contemporary, so there are going to be some noticeable differences.
So more in the xenobiotic side, presence of exogenous metabolites.
Yeah, so those samples might not be appropriate based on the context, yeah.
All right, excellent, thank you. All right, let's hear another response for if you think QC redundancy is important in your study, and if so, why? Or if not, why not? Everyone is deep in thought because it's such a really good question. And those of you who are scratching your hair and adjusting your glasses, be careful because I may actually call on you. Yeah, I'll repeat the question. So the question is, do you think that QC redundancy is important in your study? Why or why not? All right.
So I would say yes, and I'll give you 2 examples. The first we've already heard from Tracey, that if you have redundancy, then you can use it to assess experiments that would otherwise have to be junked. And we have also found it exceptionally useful for having an independent QC sample to test our bioinformatics methods. So anything you do for batch correction or anything, it massively improves your assessment. The big issue is always cost.
Yes, we've got another response here. We got two more, actually.
I'm actually, oh, sorry.
No, I'm calling on you.
Oh, okay.
Yes, ma'am. Go ahead.
So I'm actually a younger investigator, but I actually really wanted to make a case for B in the use case scenarios. Because when we are evaluating what kinds of QC samples we want to include in our runs, it gets very confusing as you start thinking, do we want to capture technical variation, biological variation, all of the different kinds of considerations for our QCs. So I can't say how we use them necessarily, but I just really want to say it's very important to me to see how you folks are all using it in the community, and especially these use case scenarios really help solidify how those QCs and reference materials really actually make a difference, especially because we all know run times can get long, and including too many QCs can be confusing sometimes.
Absolutely. Thank you so much. Yes.
Yes. To answer the question outright, if they are important, I would say yes. In our case, in my case, mostly for system suitability, just because I'm working with zebrafish, and getting reference materials in the form used in the study presented a bit ago is close to impossible. There are just no reference materials for those matrices. That's why it's mostly just system suitability with general reference materials.
Yeah, absolutely. Okay, thank you. All right, one more response on this question, yes.
Okay, that's great. So I think to add to A, when you're doing really large studies with like thousands of samples, it's really important to plan very meticulously in terms of, your pool QC, because you want it to last for a very long time. So when you're talking to a consortium or, planning how those samples are collected, make sure, there's enough for not only your sample extraction, but to pool those samples and having a really good plan for that ahead of time. We just ran a TEDdy study, which is looking at type 2 diabetes in children. And it was like about 14,000 samples, and it took a year to run, but we had several different batches. And then when you put in the, time that it took in terms of instrument time and when they're down and all those things, you really have to pre-plan your pool QCs, which is very important.
Absolutely, absolutely. Yeah, the amount of time to put in front. Okay, I'm going to allow it. One more response to this question.
Kind of a different viewpoint in terms of looking at the value of the QCs. It's what problems or sources of errors we're trying to correct, right? A pooled sample does something very different than a reference materials does something different than, say, a solvent blank versus a regular blank. So in reality, if we're going to correct for all those sources of error, we need all those sources of QCs, at least my opinion.
All right, excellent. Thank you, guys. That was an excellent series of responses. I'm going to pull out a couple of things because it's going to lead into the next follow-up question. So, and you actually saw in the presentation, there was, we already gathered information about barriers, but that was a small sample set, and we have more people here, and we think this is really important. And just in the responses, cost and availability, which are two of the biggest barriers, came up. So I just want to ask you guys, you now have the low-hanging fruit, so feel free to use that. But what barriers, in your experience, what barriers inhibit reference materials use? See? You guys, he's a quick learner. We're going to ask you to elaborate on that, sir.
Yeah, cost. And I guess maybe just as a response to the other question about their utility or their case use scenarios, we use it for accuracy determination to cross-platform comparisons. So unfortunately for NIST, at least for the polar metabolites, there's not an extensive range of metabolites. They are often measured by different platforms themselves. So, but it does provide us some reassurance that we have some mutual agreement. So that's the primary use we use it in our case. But again, it's limited in scope in terms of what metabolites are, say, measured by validated methods by NIST. So it's, in the lipids case, I guess we have the harmonization study that we can rely on as well, but that's also limited in scope.
Yeah.
But I think cost is one issue, and again, in that case, it's primarily an American sort of population. So again, it might not be appropriate for other, population groups, perhaps.
Yeah. Okay. In response to that, we have under a minute. I'm going to get Chris Beecher a microphone, and I'm going to ask Tracey if you wanted to. Hold on one second. Oh, you're going to get to talk. Just wait 1 second. Afterwards, do you want to say something about this SRM survey? Okay. All right, go ahead, Chris. I'm sorry to make you wait.
No question. Cost is probably the reason most people don't use it. And yet they're forgetting that the majority of the cost in any experiment is the time involved in working up the data. The criticality of having this material in there is to exactly save the 14,000 injections so that you don't have to do them twice. You can't, they're irreplaceable samples. And so cost is a terrible problem, but it's also a critical answer. And I just couldn't let that go by.
Thank you. All right, I'm going to just turn it over to Tracey real quick to make a little plug for.
Sure. I don't know if this is on, but so SRM 1950, we're trying to start to develop the successive material for that. So we have a survey that is out. I have a poster. I can't remember what number it is, but find me today or tomorrow. And we need your help, input of what this next material is going to look like. So it would be wonderful to have your all opinion of what that looks like, because we want it to be very helpful for the community, and we want it to last as long as 1950 has lasted now. So your help would be very valuable. Thank you.
All right. All right, next I'm going to turn the floor over to Dajana, and she's going to talk to you about the next topic.
Okay, good morning, everybody. So we'll switch gears a little bit and talk about the results from our interactive forum on data quality review. So just before I start showing you some of the results from this forum, I want to define a couple of terms that we defined for this forum. So we defined analytical batch as a series of continuously analyzed samples. We defined internal standards as exogenous or endogenously labeled compounds that we are adding in order to evaluate quality. And then quality control sample or QC sample for this particular forum was any of the different types of QCs that we can have. So it included everything from pooled QC samples, reference materials, solvent and extraction blanks, phenotypic QCs, and so forth. And if you're interested how frequently different materials were used during data quality review, I show you those numbers in percentages in brackets, and you can see, not surprisingly, pooled QC samples, reference materials, and solvent blanks were widely used by the community. Next, we can define two different time points at which we may do data quality review. So we could be checking our quality as the samples are running. So this would be defined as real-time data review. And the types of questions that we want to ask here is batch running well? Are there any issues that we need to resolve? Do we need to take action? Do we need to stop the batch? And most commonly here, right, you are trying to identify problems early rather than running 1,000 samples and then figuring out there's a problem. problem, you want to be able to react. And usually this is done either using internal standards or selected metabolites because you can check all the data on the fly generally. In post-acquisition data review, the question that we're asking there is different. The question there is, are we going to accept the batch and proceed with further biological and statistical interpretation of the data? What criteria should we use to accept or reject a batch? And then also on sample outliers, are all samples of good quality, or do we have some outliers that we need to exclude from further interpretation? So here in post-acquisition, we are really looking at holistic data review, and this is where we want to look at all the data, not just selected metabolites generally. And when we ask question of the interactive forum audience here, majority was using both. So 80% were using both real-time and post-acquisition, and only 20% were only using post-acquisition. The next question we ask is which parameters are typically being assessed either during real-time or post-acquisition data review. And what you can see here, internal standards are most common with 95% of respondents using those, followed by internal standards in real-time and monitoring TIC in real-time among the activities we do in real-time. And in terms of post-acquisition, people were looking at numbers of features or metabolites they are seeing, or also checking the performance of selected metabolite features that they know should perform well. Then we ask the question of what kind of metrics do you use to evaluate data quality. And you can see here in real time, retention time drift was the most often checked, followed by signal response. If we turn our attention to post-acquisition checks, these include everything from retention time drift, mass accuracy, signal response, and also checking both intra and intra-batch effects. And you can see all of those responses are anywhere roughly around 70% of our respondents. Then we ask what are the typical acceptance criteria that you apply to these different types of metrics. And you can see here the most common answers were maximum intensity and maximum retention time variation. So these were used by about 88% of the respondents. And this was followed by the tight clustering of QC in principal component analysis. So to show that technical variability is less than the biological variability of the samples that are being evaluated. You can see some other strategies such as batch correction, checking signal to blank ratios. Those were used by about half of the respondents. So then we ask next series of questions to really look at batch failure. So how are different labs accepting or rejecting batches or samples, and what do they do in case of a failure? So in terms of batch failure, most common strategies that were being used were clustering of QCs in PCA, or determining the number of QCs that fall within a specified acceptance criteria. And the figure on the right-hand side shows an example where the dots in red are showing a series of QC samples collected for that study. Dots in blue are the biological samples, and you can see the technical variability there is much lower than a biological variability. For sample outliers, we saw three main types of approaches. One was using principal component analysis, using hoteling ellipse, and looking at samples that fall outside of the bounds of hoteling ellipse. And other labs were using internal standards, either using quality control charts, using an overall RSD, so internal standard performance has to be less than 15% RSD across all samples within a given batch, or using statistical outlier tests as well. So after that, we started a discussion in the audience to see, to gather more information about how do different labs decide whether QC is within a specified tolerance or not. Do they use control charts or other criteria? And here we saw a breadth of different approaches. So for example, one lab starts with the principal component analysis. Then they look at the number of features or metabolites that they see in that batch and compare against their historical data for that matrix. And then they evaluate CV intensity metrics and make sure that that looks good. Another lab usually starts with looking at median RSD and spread of the RSD values. They are a little bit worried about using solely PCA because in cases of high biological variability, then even tight clustering of QCs may not show that QCs have a good performance. And they were really liking to use D ratios because they can also compare performance of a metabolite within a batch and across different batches. And these D metrics can be reported for each metabolite. So we really know what the quality of the data is. And if you're not familiar with the D ratio, so these D ratios can be calculated as a standard deviation across all QC samples for a given metabolite divided by standard deviation of that metabolite across all biological samples times 100%. And then a third lab was using a combination approach where they looked at PCA clustering, they looked at the CV value spread and median, and they also used Euclidean distance again as another objective measure of how good that clustering of QCs is. In addition to that, we also during this forum heard a lot about the need for the redundant measures to be able to rescue or troubleshoot batches. So one lab was reported that in addition to using pooled QC samples, they also use system suitability testing, and they use separate internal standards, one that was spiked during extraction to monitor for extraction issues, and one that was spiked just prior to LCMS analysis to identify misinjections or injection volume issues. And then they were also doing some real-time checks of their pooled QCs to make sure everything is running well during each of their batches. So overall, what we heard from multiple labs really is this redundancy is really helpful when it comes time for failure analysis. And there is a need for fit-for-purpose strategies. They were employing slightly different strategies depending on the type of study, the size of the study, whether it's like something like 14,000 study we heard about, or a much smaller one batch study. Then we ask different labs, what do they do when their QC samples fail? And you can see here, majority of the respondents will accept the batch up to the last acceptable QC. And then this was followed by reanalyzing any of the failed QC samples in a different batch, and any bracketing biological samples. You can see in terms of reanalyzing whole batch, about half of the respondents was doing that. And occasionally, some labs would just reject failed samples and not reanalyze them either due to limited sample availability. Then we wanted to see what was the most frequent way to detect outliers. And you can see here, hoteling ellipse was the most predominant, followed by the acceptance criteria based on standards or statistical outlier test, again, usually based on internal standards. And then for the last part of the forum, we also wanted to see what kind of data processing or data manipulation strategies were being used to ensure only good data quality was being used for further biological interpretation. And you can see here, the most common was CV filtering of features, where typically cutoffs of 20 or 30% RSDs were used for intra-study QCs. Normalization and frequency filters were the next most common, with about 71% of the audience using those. You can see a little bit surprising in terms of injection run order effects. That was done a little bit less, with 41% looking at that. Another common strategy was if there was any bad QCs, any samples that were within those QCs would be excluded, with 59% of audience excluding those. And you can see other strategies like retention time correction or scaling were less frequently used, with 29 to 41% users. So then we ask what decision points drive these steps. And in general, again, this was very study dependent. So for example, in a C. elegans study, because the metabolome was not very well known, they decided to use a more stringent frequency filter where the metabolite had to be present in all biological samples, so 100% frequency filter. In another example, they explained that they target 30% CV filtering, but in general, their good quality features should have much better CVs than this. So this 30% is not really a target, but kind of the maximum that can be allowed. So to recap, the main findings from this interactive forum were really that the QC processes were refined in labs over time. So I think following up on what Bob was saying is after finding failures or causes for failures, additional QC strategies were added to try to address those, and when possible, those were included as real-time checks to make sure everything is running okay, and the use of redundant practices, as we saw also from Tracey's presentation. So then we can switch gears to our next polling question. And here for this polling question, we will ask you guidance for data quality review in untargeted LCMS-based metabolomic studies in upcoming mQACC guidelines should cover. And this is one of those choose all that apply questions, so you should be able to select any of the answers that you think are important. So A, considerations in determining acceptance criteria. B, batch failure outlier analysis, including use case scenarios. C, metrics and reporting guidelines for data quality review. D, data manipulation strategies to improve data quality. So these are things like feature filtering or normalization. E, other. And F, I do not think that we need guidance for this topic. So hopefully the poll is running and you should be able to choose all that apply. You can only choose one? No. Okay. Crisis averted. Maybe 30 more seconds. Okay, so 30 seconds. If you haven't had a chance to answer, please answer. If you joined us late, the poll is under the conference app. If you go to workshop 9, and there should be a poll option there. Okay, I believe the poll is now closed, and I will pass the floor over to Jonathan for the discussion on this topic. Thank you.
All right. Thank you, Diana. Thank you, Krista. All right. So you should be able to see the results in your polling app now. I don't think you could have been more even across all of them. So I don't, I take that as we're doing a good job writing these questions, okay? Maybe it's a little boring on the live polling side to see that, hey, everything is important here, that's great. Y'all are doing a fantastic job, and I appreciate you hanging in with us for this long. We're gonna spend a few minutes talking about guidance for data quality review, and as normal, or as usual, we have our microphone runners set up in the back, so be prepared to throw your hand up as quick as you can as soon as I ask this question. How do you ensure, let me preface this by saying, I believe that a number of you have already been answering. I could probably pick out who would raise their hand for this question because there's some overlap here between this and the last topic. But how do you ensure data quality over large cohort studies run in multiple batches? I think one of the people that answered this before, go ahead and give her a microphone, has left the room. No, she's not. She's still there. Okay. All right, you know who you are. All right, go ahead, Jennifer.
So we try and ensure data quality over large batches by, we can talk about the analytical, but I would go back to planning is essential. And I think, yes, we can talk about QCs, and we definitely use these, and we use redundant QCs, but we also use other strategies such as protocols and quality checking of biobank samples, and this is an essay in itself.
Well, for an essay in itself, very succinctly put. Good job. All right. Here we go. We got a microphone on its way.
Thank you. I guess it can be a combination of the three ways that we have seen so far. So both checking the internal standards, the reference materials, and the pool quality controls, and try to combine these three ways. I just wrote a research grant about this, and I wrote that I will do in this way. So hopefully it can be a way.
Good. All right, let me read the question again while the rest of you consider your answers. How do you ensure data quality over large cohort studies run in multiple batches? Okay, we've had some excellent answers so far. We also, yes, we got one. We have a winner.
James Pike from Angelin Technologies. This is a little bit of a side, but pulling in some information that I've learned from the pharma industry, who do lots of real-time monitoring and quality assurance, quality control and stuff like that. They use reference material a bit more like a system suitability, where they've got specific compounds that provide a diagnosis of the instrument state. So, for example, compounds that show that the column is performing as well, show that the ion transmission is performing, show that collision energies are doing as expected when they're acquiring MSMS spectra and stuff like that. And it's just really interesting because they've got specific mixtures, and one example is something called Disco 7, where it's got 7 compounds in there, and each of those compounds have got a specific purpose to test the system in different ways. And I think that's something that's worthwhile thinking about because when we're throwing a reference material at a system, looking at it from a data analysis perspective is one aspect, but trying to capture the issue earlier on in the process at the data acquisition point, using diagnostic compounds and quick tests, quick reporting, I think is something that could be thought about.
Absolutely. All right, I'm gonna allow one more because everyone is just chomping at the bit to answer this question, which I love. So we're gonna take one more response and then I'll ask another question. Go ahead.
I believe that we need an inter-batch QC sample. So we have starting training our colleague who owns or will be running a big cohort while they are collecting sample, like working on either plasma serum. They should start to make pool sample so that if they worry about the fetal cycle And that's the point that they can make multiple QC aliquot for not only metabolomic study, but for all kind of study, because that's the way we can make sure that there's interbatch variation that can be considered.
All right, excellent, thank you. All right, next question. And I will preface this just by saying, this question asks about refining processes over time, okay? So as you're thinking about your response, please don't think that if you're a newer researcher in the area that your response is not valid, because this could apply if you've run one study, seen something you needed to change, and then made that change for the next study, okay? You don't have to have done this for years and years and years to be able to answer this question. But of course, if you have done this for years and years and years, we also want to hear your answer. So the question is, how have you refined your quality control practices over time, and why? And I know that every person in this room is qualified to answer this question, so there's no excuse for not putting your hand up. All right, we've got one in the back here.
I guess one of the things that we learned was when a reference material is useful and when it's not useful. So for some of our human studies, we could get initially a large amount of reference material from horse plasma, and we were running that alongside our samples. And then what you found with your PCA analysis was that horses are really different to humans. You know, they were well away from actually any of our human samples running along there. So actually, in terms of batch correction, they weren't a great sample set to be running alongside there. So we ended up spending a lot more time processing our pool QC and our reference materials that we're, you know, in terms of running standards alongside there, and then drop that horse plasma sample. So I think, you know, you can get a feeling for whether a matrix or a sample set, a reference material is going to be useful for you sort of long term going forward. We do a lot of lipidomics, and I think our other lesson has been that we find the lipid splash just incredibly useful as a reference material, just to check performance, you know, to sort of quickly eyeball that we're actually seeing those same peaks across a run, and nothing has gone disastrously wrong in the night when we're running LC. So those are the two things that we sort of picked up in the lab sort of on a day-to-day basis.
Very good, thank you. All right, I'll repeat the question and see if we can get another response. How have you refined your quality control practices over time, and why? All right.
So
Yes.
in our group where we work with tuberculosis, MTB lysates, we've only relatively recently added pooled QC samples and moved toward a shorter, it's sort of shorter runs due to certain technical limitations in our space. And they've definitely been very helpful, but generally, I actually haven't needed to really use them to adjust things more than the sort of our system performance standards. But I suppose if something goes very wrong, I've been using them to do certain types of retention time adjustment myself, but I don't think other members of our group have found them objectively that useful yet. But I think that's mostly that we're still thinking about the power of using pooled samples every time. So I guess my thought would be that there's something of an adaptation curve to like, oh, we're gonna add this, and then, oh, well, we're doing it, but maybe we're not actually using it fully. And so that guideline there to help implement further would be helpful.
I love that response, because you can get an idea of what you should be doing from the community, but if it's not useful for what you're doing, is it really necessary? And that's a great, actually, segue into our next topic, because one of the things he's going to talk about is the fit for purpose principle, and that's something that's absolutely important here. So I think I'm going to go ahead and close the discussion for now. You guys will have one more opportunity to provide your responses, so fret not. I am going to turn this over now to our last presenter, Matt Lewis, and he's going to talk to you about quality assurance.
Thanks, Johnny. All right. Good. So I clearly drew the short straw today because QA is kind of the bane of the expedient lab, right? These are all things that cost something in terms of time or in terms of actual money, personnel resource, and it's tempting to skip some of these steps, but in the end, is it really, I think as some of the people discussed earlier, is it really overall cost effective? I think to Chris Beecher's point, is it really cost effective in the end to skip things like that? So let's just talk about what is QA first of all. It's sort of the black swan or the ugly duckling of the QC QA group, right? So here, sorry, where's the pointer? Is there a pointer on this? No pointer. Okay. Oh, here's a pointer. It's hiding. Yeah, okay. So going back to one of mQACC's seminal papers here, it's nicely below the line, right? And all this nice, exciting stuff above the line, and then there's this kind of below the line. And so in preparing for this and trying to understand exactly what QA means to most people, I use this as a guide. I appreciate that there could be other things that constitute QA, and please, if you see something missing, ask. But I just took this literally because it's the foundation that we have to work from, and put these sort of here with the idea of some example considerations, right? So I think looking at it like this kind of invokes some perhaps emotion around QA. I think the first thing that we have to dispel is that the topics at the top are not the pinnacle of quality, and they don't necessarily make quality. I can very well assure you that one person in one lab with one instrument can produce quality that is of a very high standard, and they could even do it reproducibly, right? So this isn't to say that unless you have quality system audits operating monthly, that you shouldn't be trusted to produce high quality data. That's not the point. The point is really, I think, more of scale. Okay, so down here, these are more common in our field because these principles like training your staff and having logbooks, these are more core principles of just business and employment and then of basic science, right? So these are very common regardless of whether you're in a big company, a service organization, a center, a CRO, a core facility, or an individual PI academic lab. Whereas some of these at the top, these are often just less common because they're associated with often larger organizations. And here QA serves to drive efficiency in the organization. Things like liability and accountability may be coming into play here where they're less so in the PI-driven lab, where if something goes wrong, you just collar the student, they do it again, right? And that is a way of assuring quality. But there's different systems for different scales. So I just want to dispel the myth that you can't do good quality work if you don't do all of these things. It's all about being, as Jonathan said, fit for purpose. So looking across the different types of labs, these are a few, these are the ones I could think of. I'm sure I missed something, right? But we have our clinical and academic research groups, PI-led groups. Could be small, could be large, right? Usually in academic institutions. We have core facilities, often similarly in academic institutions, but they're running perhaps at a different scale, and they're a little more service-oriented, right? Collaborators come in, and there's a little more accountability there. It's not their own personal research, so if you screw something up, really you got to go back to the logbooks and be able to absolutely demonstrate that you followed procedures correctly. Then you have the argument about was it your samples or my analysis, right? And that's where liability kind of comes into play. Even more so enhanced in CROs and larger centers who do a higher volume of analysis, doing really multidisciplinary collaborations, offering services at a higher level, let's say. And then finally, we have highly regulated labs, pharma, government, et cetera, where just by nature of who they serve, the bar for quality assurance and the bar for auditing and the bar for following standardized protocols is much higher just because, right? Just because of the potential downstream impact, the decision-making that might be made based on that analysis, right? In research, the decision-making is, let's plan another experiment, whereas in a clinical laboratory, it might be, let's treat a patient a certain way. So there's different thresholds of accountability in this case across this landscape. So a wonderful archetype from our very, what I did was I polled internal to mQACC. I asked people, what are you doing? Just give me some examples. So this is a limited set of examples from people who responded positively to my asking within the mQACC organization. And I'll show you just some examples, okay? So our very own Maria here in the front row, say hi. I asked the question, which one of these things that we just kind of went through do you really feel you excel at? And she gave what I think is an archetypical answer here. I focus on SOPs, I focus on temperature monitoring, I focus on maintaining my equipment and maintaining a schedule of that, logbooks, and staff training. Things that I think are less of a focus in the typical, perhaps, academic lab, audit trails, right? Because in some cases, I don't know how big your lab is, but in some cases, the audit trail is knowing who ran the project. And that serves the purpose of an audit trail in that kind of an organization at that scale. You know that this person was vertically responsible from start to finish. And we'll see in larger organizations that's not the case, and auditing exactly who touched what becomes more procedurally important. And similarly, proficiency testing, that's an interesting one. That's when you go into the lab and make sure someone knows exactly what they're doing by some pre-planned mechanism. And I know you as a PI are doing that day to day, right? But it's not really considered proficiency testing as such, okay? So this is a nice adaptation. And Maria gives us really nice examples here about why this is happening. And these are critical. We have to understand it, right? Number one, we want to be accurate. Number two, we want to be reproducible. These are really why an academic lab would be investing in QA strategies, not being the expedient lab that doesn't do any of these things. Nice response from a colleague at TMIC here. So this is an organization, I think, a little more service-oriented, working now to ISO certified methods, right? So they are ISO compliant lab. They have a nice quality system here with actual QA and node managers, right? Actual personnel in place to make sure that whatever is discovered the data that's generated is checked. Anything that's found wrong is acted upon. The systems are planned out and they're enacted so that data can be checked, and it's this nice kind of QA cycle, right? So you can see this is a little more formal. There's overhead in doing this, right? I discussed with a colleague last night who's starting to work towards ISO certification, and it's a pain, right? The expedient lab wouldn't do that, but some labs who want to access certain types of customers or collaborators this will be worth their while. Not everyone's while, but some. And this group also has their own in-house developed electronic quality management system. So that's nice. Some groups invest in that, but I'll show you some other highly regulated environments where that's not necessary to give you some hope that you don't always need these very fancy solutions. A third kind of example here is BASF Metabolome Solutions. They, I think, similarly have these types of investments across the board. Here, they're good laboratory practice certified, right? So these certifications start to become important, especially if you're providing services. If someone is going to come back to you and say, I trusted you with my samples, I trusted you with my precious materials, and I want to know exactly what happened. Now you're working to a different level of accountability. Not necessarily a different level of quality, but a different level of accountability. So the assurance of quality in your organization has to be slightly different. Okay, so from my colleague to the left, Jonathan Mosley here from the EPA, works for the government, and has electronic notebooks in OneNote, totally accessible, okay? So I want to make the point that it doesn't always have to be fancy stuff. For his equipment maintenance, it's right in OneNote, and this is how Johnny runs things in the EPA. This is entirely achievable by everyone in the room, no problem. So don't think that solutions have to be these fancy in-house things. For temperature monitoring, right, freezer monitors are very, very common. So this is an example that Jonathan sent from his work at the EPA. Pantelis Takis from my former group at the National Phenome Center sent this through, where they actually monitor room temperature and then store that data alongside the analytical data and use it to understand the analytical data. So actual integration of external temperature monitoring. This was, I think, one example of that goes a little further than most there in terms of actually integrating and using that data. SOPs, Oliver Fien from his group was very kind and sent some slides related to how he considers SOPs. What makes a good SOP? It's the inclusion of all steps, a pictorial workflow, all reference materials, how to do data analysis. Everything is in one place, right? So you know if you're going into the Fien lab, you're going to be resourced with decades of knowledge, and they're all documented. This is certainly good for driving efficiency at any scale, right? Even if you're a PI-driven small lab, this means efficiency for the next starter. You don't have to train them up from zero. In the interest of time, this is, again, from my former group, thanks to Pantelis Takis. Right before I left, we took a democratic vote in terms of what do we do with all these SOPs that we have, and they decided to make them all public, right? So these are all now public on GitHub, along with all the annotations that you can find with these SOPs. And making these things public, we felt was very important. I think many groups feel is important. I remember when we were starting out in this endeavor, seeing Rick and others in the room work from Husermet. And all their quality control and SOPs were all online, all right? So we could learn from. So this is kind of standing on shoulders kind of stuff. But do you make them publicly available? How are they closed within your lab? Are they intended for the benefit of others? These are all important considerations. As we get further up that pyramid, we get towards more difficult topics, proficiency testing. So this is from Annie Evans at Metabolon. In terms of exactly what they do and why they do it, the idea here is specifically to routinely make sure that you have a staff, it could be a staff across sites in a very large lab, who are behaving appropriately and competently in their ability to make measurements. Again, this is perhaps more intrinsic in smaller labs, but as it becomes larger organizations with more liability, it has to become more explicit, right? And that's what Annie's talking about here. Similarly, with audit trails, you may walk out and talk to your students and say, where's your notebook? I want to see your notebook. At an organizational level, the idea is this. And it really, I love this particular word. Audit trail is important in troubleshooting issues, but also in defending work performed. So you can start to understand why formalized audit trails are important, because someone may come back and say, you provided a service, and I want to know exactly what happened, and the company may have to defend, right? Finally, quality system audits. In the EPA, John has a nice kind of system here of plan what's needed, do it, check that it works, act, and this cycle goes around in terms of following their QA documentation, planning it, reviewing it. They're doing external audits every 3 years. I think at Metabolon, somewhat substantially more often, but for different purposes. So again, auditing a very large or very regulated organizations becomes a specific challenge that looks very different from the common academic lab. Okay, so that's a quick spin through QA. Thanks to all the people who very kindly sent me some material to discuss today. The reason this talk doesn't look like the others is because we're just getting started with QA in mQACC, okay? So if you're interested, there's a new task group here. Don't confuse QA with QC like I've clearly done here on this slide. It's very easy to do, but bottom line, QA assures data quality. It doesn't create it, it doesn't make it, it just helps assure it for efficiency and make sure that things are fit for their intended purpose, right? So that's it from me. I will go on now to the next polling question. I missed the rehearsal yesterday, but I've now seen my colleagues do it 3 or 4 times. So I think I can nail this. So everyone have their apps open again? Thank you. We will go. So guidance for quality assurance and untargeted LCMS-based metabolomic studies in upcoming mQACC guidelines should cover, and this is another infamous choose all that apply. Hopefully that's true. I'm seeing thumbs up, so I think that's true. Considerations in adopting QA practices that are fit for purpose is A. Descriptions of tiered QA approaches including use case scenarios, B. Representative QA templates like logbooks and protocols, C. D is other, or E. You don't think this topic should be included. while we're waiting, I will comment that every time the responses to these questions are even across the board, it means that mQACC has a lot more work to do. So, so you may hope that one may win out over the others, but I think what we're seeing is there's just a lot of work to be done across the field. Okay. Oh, Krista says we're still getting responses. Please take your time. All right. 30 more seconds, you heard it. All right, that's it. Thank you very much. I only had one question, so I think that's it. Yeah? Good. Thanks for your time, for listening. I turn it back over to Jonathan.
All right, thank you, Matt. And for the one who drew the short straw, you performed admirably. So. Okay. So hopefully you're seeing the results of the polling on your screen. And once again, you guys have performed admirably and evenly. So it seems like of those first three options, considerations in adopting QA practices that are fit for purpose, descriptions of tiered QA approaches, including use case scenarios and representative QA templates, are all important to you. And that's fantastic. That's great. That's a great confirmation that we're on the right track. In the time that we have allotted to us, I want to do two things. Of course, we're going to discuss the quality assurance polling result just like we've done all the others. And then given the hiccup at the beginning of the workshop, if you can remember back that far, after we discuss the QA topic, we'll go back and reopen that first polling question about the living guidance document as it relates to quality of metabolite annotation. So first things first, I'm going to ask a follow-up question on guidance for quality assurance. And this question stems from the fact that, as Matt mentioned, for all the other topics besides QA, we held interactive forums that were external to mQACC, whereas by the time we got to quality assurance, mQACC was large enough, we were able to do an internal poll. Now this is the opportunity for you to, whether you're in mQACC or not, for you as a member of the community to provide feedback on the proposed approach to QA that we have just presented to you. So the question is, do you agree with the proposed tiered approach to QA, and why or why not? All right, so we've got our first taker. A microphone is on its way.
I actually had a question about what that meant, because I was curious too, if there was like a minimum or a universal thread in QA, QC practices, because it made a really great point for the fit for purpose. But did that tiered approach basically, was that what you meant by like the first tier would be like this is the minimum, and then additional tiers are your fit for purpose?
I'm going to let Matt answer that question.
I think. Thank you. Oops. All right. Make a bunch of noise first. I really, having contemplated a lot of the material that was sent to us recently, I would struggle to really put into effect a tiered system, frankly. I would struggle with that, just because the more I think about it, the more it comes down to how formal it is, how intrinsic it is. I think every lab kind of does all of those things, right? And if they don't, they're going to have very poor data quality. But I think generally labs do. It's just to different levels. So I mean, there are intuitive tiers like train your staff and have SOPs, right? Those are things that it's really kind of hard to do without, and that would be formulate a pretty basic tier. Data monitoring is probably, if there were a middle tier, I would say lab temperature monitoring, freezer monitoring, and system performance monitoring. Those are all next up, right? For sure, bottom tier, you train your staff. Next up, if you're a professional lab, you want to monitor those things. And then a third tier could be some of the things that go along with only certain circumstances, auditing, ring testing. Sorry, I meant proficiency testing. That was Freudian, I guess, in a way, because the academic version of that is ring testing, right? So I would put some of those things perhaps in a third tier, but it's a bit of a stretch to do so.
All right, we've got a microphone on the way to you.
Hi, I'm Gonzalo. I'm from the IBBR. And I think as a community, we need to be a little bit careful how we do the fit for purpose. So the QA system is already fit for purpose, right? So it's been developed, it's been applied to industry, to very specific applications. So anyone that has gone through a QA, ISO 17025 knows that it's fit for purpose. So you say what you're gonna do and you're audited against what you say you're gonna do. So I don't think we can apply a QA system and just choose things not to use. So we do need to follow the whole tier of the QA system and just say the limitation. So there's consequences for not doing an audit, right? So we need to be aware of that. And an audit doesn't mean that you get some company external to come and audit your research lab. That's not what we're talking about. It might be just grab your PhD students and say, today we're gonna do an audit of the lab and do an audit of all the protocols and the inventory and chemicals and so on, right? So I think there's, the QA system is already very good and very well established. We just need to tone it down for the purpose. But we can pick and choose what parts of the QA system we use or implement.
Thank you. That was a very thoughtful response, and it's got all of us thinking right now up here. All right, would anyone like to answer? I'll read the question one more time. Do you agree with the proposed tiered approach to QA? Why or why not? And really, there is a related question. I'll read it too. Is the fit for purpose principle appropriate for QA? Why or why not? And Gonzalo just definitely predetermined that question should be asked. So thank you. So I'll repeat. Is the fit for purpose principle appropriate for QA? Why or why not? Yes, Jennifer.
So I take a very pragmatic approach here, that first of all, what do we mean by fit for purpose? You could take a lab that does absolutely none of these things, doesn't check their data, and publishes results, but they're meaningless, but nobody knows that they're meaningless. I would argue that's not fit for purpose. So we should agree that everybody needs to be doing some form of QA at some point. But this idea of the tiered approach, as I'm thinking it through, I think it's less about what you do, but how you do it. And then we get on to the fit for purpose. We have to consider the cost and time implications. And if we don't, then people are not going to adopt the practices. That's my pragmatism.
Thank you. We appreciate your pragmatism, Jennifer. Yeah. Oh. Okay. All right. I think we have time for maybe one more response. Yes.
Thank you very much for the webinar and for the workshop. It's Antonia Garcia from Sembayo in Madrid, Spain. I'm an audit expert in ISO 9000. And of course, for quality assurance, the first is the internal assurance. Then, how can we prove, how can we have evidence of the assurance in your lab? And then we can think about the second stage, that is external assurance. And perhaps there could be a kind of or experiment of organization in order to have evidence, because assurance is always to have evidence of that you are working according to the certification of the criteria that was previously given. But first thing is to establish what will be that criteria. And for untargeted LC-MS, it's quite, this is the first challenge here, of course. And for sure, I think always should be a kind of agreement of organization between different labs for establishing this. We can establish an agreement, for example, for lipidomics in plasma. It's an option, or for any other tissue, for example. Or we should organize this according to the technique and the retention mechanism, and of course for the matrix. Some kind of, this is my approach. Thank you.
All right, thank you. Thank you for your response. All right, we are going to wrap up the discussion time now because we're in the last few minutes of our workshop, and we're going to ask one more thing of you. We need you to answer another question in your polling apps. So go ahead and pull out your apps. We're going to reload that first question that should have been choose all that apply, and now it will be choose all that apply. So while you guys are getting that up, bear with me 1 minute while I scroll back through too many slides. Okay. You should be able to see it in your app and open it. I'm just going to try to get to, oh, and here we are. So the question is guidance for the quality of metabolite annotation and identification in untargeted LC-MS-based metabolomic studies and upcoming mQACC guidelines should cover considerations in choosing quality criteria, types of evidence, including use case scenarios, evaluation metrics, reporting guidelines, other, or I do not think that guidance for this topic should be included. And please bear in mind, the first time you answered this, you were unceremoniously forced to choose your best option. This time we want to know what all you think are that apply. So please choose all that apply. All right, we're going to let it go for another 30 seconds. While you're putting in your last minute selections, let me just say once again how incredibly excellent of an audience y'all have been. I know we have thoroughly enjoyed this up here, and we hope that you have too. Okay, all right, we're going to go ahead and close the question. And then we will share the results live for you to see. All right, and at this point, we're not going to introduce any specific topic or question. We only have 5 minutes left in our workshop. So I just want to open up the floor. There's been a lot discussed. There's been a lot of information presented to you. If you guys have anything that you would like to comment on, ask a question about, this is the time to do it. Yes. Okay, could we get another microphone over here? You sir in the back, go ahead.
I think obviously community consensus is really important, but community practice is even more important. I work in industry. I get audited at many levels. So how as a community can we improve adherence to a community standard? What are the things that we can do?
I hope you weren't expecting me to answer that question. Okay, good. It's a good one for provoking thought, though.
Yeah, hi, I'm Pim Lennarts, Vrije Universiteit Amsterdam. I want to come back to the reference materials because as we discussed it, it's mainly quality control. But can we not build also on annotation? Because a lot of people using the same reference materials, so we do our own annotations every time. So can you not build on a website or whatever that we can put our annotations there so that we are building together on an annotation library, more or less?
Yeah, so we're actually, I guess you could say mQACC is piloting a way to do this. I mean, let me take that back. This has been done before in mQACC, but we are currently doing a survey on your identifications and annotations in NIST 1950 as a reference material for the purpose of obtaining consensus values. We could certainly take that, use it as a jumping off point, and open that up on mQACC's website as you tell us what reference material you're using and give a, we have the format and the structure there to gather the information. I think it's a great idea.
Right, thanks.
All right. Oh, yes, let's get a microphone over here while Tracey comments on this.
I'll just quick say that in the 1950 survey, we're having a hard time having folks provide their data for this. So that's another stumbling block that we have to think about.
And then
Yes.
I had a question on annotations, because machine learning methods are starting to be used to generate annotations, and those seem different to me than using a reference database or an authentic standard. So I was wondering if there was going to be any discussion around how you report your guidelines or your quality criteria for machine learning generated annotations.
That's a great question. Rick?
It's a great question. Is it on? Yeah. It's a great question. It's difficult. It is annotation. It's not identification, even because it's machine learning within there. But there's discussions at the moment about what sort of level of confidence you have in those as well. So it's something that's been discussed within the Metabolomics Society task group on metabolite identification.
All right, we are in the last 2 minutes, folks. I am going to meet Arpana after the workshop, and I'm going to give her the muffin. You can't have the muffin, Krista. Sorry. Yes, go ahead.
Just to the gentleman whose question a couple questions ago, I just want to point out that we do have a reporting standards task group within the group, and that is one way of driving some adherence, right? But to make the point, it's really tricky because no one likes to be told what to do. And you have to be careful as a standards organization. Everyone in the group is conscious that no one wants to be heavy-handed, and that's totally against the principles of what we're trying to do, but build community consensus and then ultimately enact it through perhaps through reporting standards is one way forward.
So I'm just going to follow up on this. If anybody's interested in the reporting standards group, come and see me. No, we don't want to tell anyone what to do. We want to nudge and advise. That said, if you're reviewing papers and you feel that the paper is making claims without providing sufficient quality assurance and control evidence in that paper, then we are strongly encouraging you to be asking for that further information to be provided. I think that's basically quality assurance at an article publishing level. That should obviously be dependent on the grade of paper.
All right, we are officially out of time. I need to release you all. I don't want to, but I must. So thank you guys so much for coming out. And if you see any of us throughout the conference, please come up to us, ask us questions, shake our hands. We'd love to talk to you. All right, thank you.